All right. Welcome to the Revolution. And we have Galen Martin with us today. And Galen is a postdoc in Leach lab, and he will talk about his work related to transposal. Galen, please take away.
Sure. Yeah, hi everyone. Thank you for coming and listening to this and for the opportunity to talk. Just to introduce myself a little bit, I'm Galen Martin. I was a graduate student in Brandon Gott's lab at UC Irvine. I graduated in a couple of years ago, and this fall I started as a postdoc in Damon Lish's lab here at Purdue University. Um, I'm going to be talking about secondary structure and transposable elements in
Yeah,
maze, which
I'm
is,
sorry. I
oh,
don't see the slideshow yet.
oh, you don't. Okay. It's weird. Am I sharing anything?
I don't know.
Uh,
I feel like I'm seeing a picture of corn and fog in the background. Uh,
yes, that's what you
yep.
should be seeing.
Oh, okay. I'm sorry. Go ahead.
Oh, no worries. Cool. Um, yeah, let's see. Change this. Cool. So, as I'm sure everybody here probably knows, the maize genome is primarily composed of transposable elements. They make up about 80% of the maize genome. And from sort of a, abstract view, transposable elements and genes are sort of the same type of thing, right? They're DNA sequences in the genome, and the autonomous transposable elements, at least, follow the same pathway, essentially, that genes follow, where they're transcribed into mRNAs and then translated into proteins. And of course, the real difference between genes and transposable elements, as we understand them, is that genes go on to do important cellular functions generally, whereas the proteins created by transposable elements go on to transpose the transposable element from one locus to another and duplicate themselves. And of course, this causes a bunch of problems for the host genome. So genomes like maize have evolved a bunch of machinery that's designed to stop this process, to keep transposable elements from transposing. Um, and they do it really effectively through a couple of different mechanisms. Firstly, they do transcriptional gene silencing of those transposable elements, where they put repressive epigenetic marks onto the transposable element DNA sequences that prevent transcription and keep this process from happening. But they also silence transposable elements when they are transcribed post-transcriptionally through RNA interference. And in this pathway, the transcripts are broken down into small RNAs, 21 to 24 nucleotide small interfering RNAs. And the sort of neat, elegant thing about this entire mechanism is that these two methods of silencing feed into one another. So the small RNAs produced by RNA interference can go on to direct methylation and repressive histone marks to the complementary transposable element sequences in the genome. And these highly methylated heterochromatic portions of the genome also recruit alternative polymerases that feed and create these small RNAs. So there are a bunch of these sort of overlapping mechanisms that ensure that these transposable element sequences are silenced. And whenever you look at the maize genome, the modern maize genome, even though it is, mostly transposable elements, pretty much all of those transposable elements are silenced through this pathway. But although these two mechanisms feed into one another, none of that really explains how the process actually starts, since one of these two mechanisms is essentially required for the other mechanism to begin. And it's also unclear, since, as I said in the beginning, at abstract level, transposable element sequences and gene sequences do the same types of things. it's not always really clear how transposable elements are distinguished from genes in the first place and how they prevent genes from being fed into this same pathway and silenced. There is some evidence for how this can occur in the first place. We know that RNA interference can be initiated by double-stranded RNA. It usually is initiated by double-stranded RNA that gets fed into these dicer enzymes and then cut up into small RNAs, which leads to silencing. But whenever you have a sufficiently structured RNA, an RNA that folds in upon itself and creates a bunch of double-stranded RNA essentially through a hairpin, that can also feed into this pathway. And one of the few examples that we actually have of initiation of silencing of an active element comes from Damon, my current advisor, who in the 90s discovered this particular locus called mu killer. And mu killer is interesting because it heritably silences a particular transposable element called mutator. And what this scheme here is showing is in A, we have the full mutator element, and in B, we have the mu killer locus. And the mu killer locus is essentially derived from the autonomous mutator element. And it's experienced a structural variation with a deletion and an inversion that causes it to, whenever it's transcribed, create this very, very long hairpin that then goes through that RNA interference process and silences the active element that it was derived from, mutator. So this is mu killer and mutator. And here you can see a segregating ear that's segregating for the mu killer locus. And you can see here in this kernel, this is a mu active element with a reporter gene that makes it, show this coloration. This is one that has mu killer, and you can see that even though it has that formerly active element, it's being silenced and not showing that coloration. And this one has no UDR. So you can see that it's heritably silencing UDR. And this is very similar, really, to the process of microRNA generation, where you have these genes that produce pre-microRNAs with long hairpins that go on to create microRNAs that then repress expression of genes. So the question was raised, sort of the impetus for this project was raised by this paper from our collaborator, Alex Busios, who is studying these LTRs, these LTR elements, transposable elements in maize. And what he found was that these LTR elements have these long palindromic repeat regions within the LTRs of the elements that could potentially create hairpins like the microRNAs and like the mu killer locus that silence the elements themselves. So what they did was they sequenced small RNAs in maize and mapped those small RNAs to the long terminal repeat loci of these particular LTR elements that they were studying. And of course, this is just a cartoon version of what this looks like. In reality, the small RNAs are 21, 24 nucleotides. But they found that in these particular palindromic regions, there's a high level of mapping of these small RNAs to those regions. So they could infer from this that there's something about that particular region that might be initiating silencing through this mechanism. And here they're showing these dotted lines. This graph represents the long terminal repeat regions of these elements. And the dotted lines represent the palindromic regions of those LTRs. And these are different lengths of small interfering RNAs. And you can see that these palindromic regions are hotspots for these small RNAs. So the idea was that, these palindromic repeats fold in upon themselves and form a substrate for RNA interference. So what we wanted to do with this project was try to extend this model and see if it could be, a general phenomenon among transposable elements and other parts of the maize genome that experience high degrees of methylation and small RNA production. So we came up with this self-priming model of de novo silencing, silencing of active transposable elements, where it's possible that transposable elements produce transcripts that form hairpin structures, and those hairpin structures initiate small RNA silencing. This is just a cartoon, right? The DNA gets turned into a transcript. That transcript forms a hairpin that acts as a substrate for production of small RNAs that then goes on to silence the locus that they came from. So you might ask, why? Why would transposable elements produce something that's going to eventually cause them to be silenced? There are a few potential reasons, right? It could be that these structures are necessary for transposition of the elements themselves. This is a picture of my friend James fixing a gill net, which is a type of net that's used for catching fish, right? And as you might infer from the name gill net, the way that it works is the fish has gills. The gills get caught in the gill net, and you pull the gill net up and you pick the fish out of the net. The fish don't have the gills because it's going to get them caught in the gill net. The fish have gills because they need gills, right? And the fisherman just takes advantage of the fact that they need those gills to catch the fish. Could alternatively be the case that Ts actually benefit in some way from facultative silencing, and we'll get more to that in a little bit. But the sort of big questions in this project were, how do these distributions of secondary structure actually look across different types of elements? And do they always correlate with small RNA abundance? And how might this model affect genes? Because genes also have some degree of secondary structure. So this is a sort of a scheme of the general types of things that we did across this entire project that I'm going to build on. But everything is based on annotations, right, in the maize genome of transposable elements and genes. And the first thing that we did is actually look for how secondary structure distributions look across these different types of elements, transposable elements, genes, and other things. So first we annotated regions of high predicted secondary structure. And we did this in a couple of different ways. Me and another graduate student in the lab both separately looked for regions of high secondary structure that would be expected to form hairpins that might lead to small RNAs. And of course, the way that we did this is based on looking for regions that form hairpins that are like microRNAs, right? They form hairpins that are at least as long as microRNAs hairpins tend to be. Sometimes I'll call these highly structured regions. Sometimes I'll call them microRNA-like regions. So just bear with me as I kind of go between those different nomenclatures. But the general idea remains the same. We annotated regions that were highly structured, that formed, long hairpins, versus the rest of the element that has some degree of secondary structure, but it wasn't quite as intense as it was in these particular regions. So the first question, how do these distributions vary across transposable elements? They vary quite a bit. So on the y-axis here, we have the minimum free energy, which is just a measure of secondary structure. The more negative it is, the stronger the secondary structure is in a particular locus. And these are distributions across all of the different types of transposable elements, as well as, and these are in the Wicker nomenclature for transposable elements, as well as microRNAs and different types of genes. And this line here represents a threshold that's supposed to be microRNA-like via a few different sort of empirical measures. as you can see, for example, the microRNAs tend to be below the microRNA structure threshold, right? There are a couple of important things that I just want to point out about this, just because they might be important later. Firstly, the most structured elements in general are the copia LTR elements, which is essentially the same type of element as Alex Busios's project was looking at, which I was talking about at the beginning of this. And you might also notice, importantly, that genes also tend to have secondary structure somewhere within that element that resembles a microRNA. So that's why we ended up looking quite a bit at genes as well. Structure also varies across the elements, as you might expect. For example, in the long terminal repeats of these copia elements, that's where you find the secondary structure. So this is sort of a sanity test, right? Where these copia elements have high degrees of secondary structure in their LTRs, but not necessarily across the rest of the element. You might also know that helitrons, these represent distributions of secondary structure within helitrons. We find that the high structure in helitrons tends to be at the 3' end, which is what we know about helitrons, right? So there's all this variation in secondary structure across these elements. But the main thing that we wanted to look at is whether the self-priming model is universal, or in other words, do these regions of very intense secondary structure correspond to high abundance of small RNAs? So we did this in the same way that Alex had done it previously, where we mapped small RNAs across these regions and looked to see if they were more abundant within them. And we did this by measuring the small RNA skew towards these regions, which is essentially, well, I'll get to it in a second, but this is just showing the basic idea, right? Our expectation is that these small RNAs map heavily to these structured regions, but less to unstructured regions, the rest of these elements. So I'm going to be talking a little bit about this measure of skew, which is just the proportion of small RNAs that map to the microRNA-like structured regions within elements. And so if skew is -1, this is really the important thing, right? If skew is -1, then all of the small RNAs map to the non-structured regions, whereas if skew is 1, they map to the microRNA-like regions. So this is our expectation, essentially, is that skew will be more positive than it is negative, right? And we can look at this again in these copia elements, and what we find is that, yeah, in all terms, all of the different lengths of small RNAs, they tend to be more abundant in these structured regions than in the other regions of the elements. We find that it's the same for copia, for these Ti3 elements and other unidentified LTRs. We also find that this is true for most types of DNA transposons as well, and there was some variation between autonomous and non-autonomous, specifically that the non-autonomous elements tended to have less skew than the autonomous elements, which sort of makes sense because the autonomous elements should be the only ones that are actually being transcribed at any point in their history, right? Now, most interestingly, the skew is highest in genes. Genes, the vast majority of small RNAs in genes tend to map to the structured regions within those genes. So even though genes tend to have less of these microRNA-like regions than copia elements, for example, the microRNA-like elements that do exist within genes tend to have this pattern very strongly. We also find that there is a, I don't know what I did. We also find that there's a peak of methylation at these hairpin microRNA-like loci compared to just random sites across all of these different types of elements. So obviously, it was interesting to us that we find this pattern in genes. So we wanted to know how this might actually affect the genes. Obviously, our initial hypothesis is that this is a way that transposable elements are identified and separated from genes and subsequently silenced. So we wanted to see what happens to genes whenever they have these highly structured regions within them. So then we looked at expression of genes with structure and without these microRNA-like regions to see how expression differed between them. And interestingly, we find that the ones that have structure tend to be more highly expressed than those without structure. So clearly, these regions aren't actually silencing these genes. And there could be a number of explanations for this, right? It may be that these genes need these highly structured regions, important genes that are highly expressed need these hairpins in order to, for example, direct them to different places within the cell or to recruit various enzymes to them for processing. But it's still kind of a strange result, right? Being that we expect these small RNAs to be silencing elements, and we don't find that that's the case. So next, we wanted to see if there was any effect in sort of a larger time scale. So we looked at variability of expression of these structured genes versus unstructured genes between a bunch of maize lineages. And of course, we used the NAM lines because they're a great data set for this type of analysis, right? And our expectation was that perhaps these structured genes, we saw that they're more expressed, and that's what this is representing, right? They make more transcripts than the non-structured genes. But it could be that these are more stable over, the entire phylogeny of maize versus these highly structured genes, which might be more unstable because there is sort of this conflict between RNA interference and expression. So we looked at these 26 NAM lines and looked for differences in expression among them between genes that had structure and those that didn't have these highly structured regions. And of course, I'm sure that everyone here is familiar with the NAM dataset, but it includes these 26 founder inbred lines that are supposed to represent the diversity of modern maize. And we looked for variation in structure, and this is a simplified version of that analysis, where this is the coefficient of variation in expression between genes that have these microRNA-like regions and genes that do not have them. And this coefficient of variation represents, a normalized variability of expression among those 26 lines that's normalized for the actual magnitude of expression across different genes, right? And we find that there is significantly more variation in expression in those structured elements versus the unstructured elements in the 26 NAM lines. So there's potentially some effect of RNA interference on expression, maybe on a larger time scale than just one line or one individual, right? So our big questions in this project were, how do distributions of secondary structure vary? And we find that they vary a lot, but most elements, whether it's transposable elements or genes, tend to have these highly structured regions. And, we did essentially two completely separate computational pipelines for trying to map these structured regions, and we find essentially the same thing, that most things have something in them that has the potential to make a hairpin that's microRNA-like. Is the self-priming model universal? Well, yes, at least in terms of the fact that strong hairpin structure is correlated with high abundance of small RNA mapping. And how might that affect gene function? Well, genes with hairpins tend to be conserved between species. I didn't really show that, but We also looked at centrologues and found that, uh, genes with the centrolog in sorghum, for example, tend to have more of these hairpin regions than genes that don't have a centrolog. But they're also more stochastic in expression if you look at them across the 26 NAM lines. So the stuff that I'm working on now, I'm still working with Brandon on some extensions of this work. And what I'm really interested in now is looking at transposable elements in a genome ecology context. I'm sure some of you have heard about genome ecology in terms of transposable elements. The basic idea here is that transposable elements represent sort of like organisms in a ecosystem where the ecosystem is the genome. And this whole idea sort of came from the fact that you find that different types of transposable elements are found in different places in the genome. Sometimes they're really close to genes, sometimes they're really far away from genes, sometimes transposable elements only colonize the centromere, and sometimes they don't, right? So transposable elements, as we've already seen here, are extremely diverse, and they appear to sort of take advantage of niches within genomes. So part of what I'm trying to do these days is extend this to a genome ecology sort of framework and understand whether this secondary structure, which is an intrinsic property of transposable elements, has some correlation with where you find these transposable elements and what their sort of lifestyle within the genome might be. So yeah, so one of the things that I've done recently is look at how differently structured transposable elements relate to differences in structure across the genome. And this type of analysis is really sort of difficult to interpret, as a lot of you who do genomics and maize know. Whenever you look across the maize genome, you're mostly looking at copia elements because copia and TY3 are so extremely abundant. But here, what this is showing is across one of the chromosomes in maize. This is chromosome 9 in maize. This black line represents the density of paired bases, which is just a metric of local structure, and this was done in windows across the entire genome. And what you find is that really this sort of landscape of structure essentially mimics the landscape of copia diversity across the genome. So really the entire structure of the genome is sort of aligned with the density of these copia elements in the genome. Remember, these are these long terminal repeat elements that I was talking about at the very beginning of this talk. But what's sort of interesting is that you find that these copia elements are in these chromosome arms where there's a lot of structure. Conversely, we found that these TY3 elements had less structure than the copia elements, and you find them outside of these highly structured portions of the genome. And of course, there are numerous explanations for this, a ton of things that could be influencing this. Yes, yeah, you're totally right. There's also a ton of diversity in terms of where individual families within these larger groups, copia and TY3 are these huge clades that include a bunch of very diverse elements within them. And of course, we're looking at that as well. But whenever you look just generally across these very wide categories, this is what you find. And there's like sort of the genome ecology explanation for this, that these copia elements, maybe they need to have higher structure because the stuff around them has higher structure. But something that's probably more likely is just that the structure of the entire chromosome is mostly defined by copia elements because they're so incredibly abundant, right? And so I guess what I'm trying to get at here is that it's really hard to actually look at this after selection has taken place and you're looking at these ancient silenced elements. So of course, now that I'm in Damon's lab, what I'm trying to do is, get around this by actually looking at active transposable elements rather than ancient silenced transposable elements, and try to differentiate these things like insertion preference versus selection that are very difficult to do with these ancient elements. So this is mostly just kind of a teaser for my current project in Damon's lab. But what I'm doing is, anybody who knows Damon knows that his system is the mutator element, and the mutator family, the active system of mutator elements, includes MuDR, which is the autonomous element, and a whole host of non-autonomous elements that MuDR also moves around the genome with it. So what I'm doing now, or what's convenient about this system, essentially, for this genome ecology type project, is that we can use really inexpensive and fast short read sequencing by doing PCR that reads outwards from the TIRs in this mutator system and mapping those reads outward into the flanking regions to map insertions of active elements. And so this is a representation of some of my data showing the autonomous elements, MuDR, and a couple of the non-autonomous, hangers-on, Mu8 and Mu7, and their insertions in one particular family. This was just an outcross between a mu-active family and a regular B73. And so you can map these individual insertions where they actually go into in the genome before a whole lot of selection has taken place. Of course, if there was an element that was completely fatal, then you wouldn't see it. But most of the selection hasn't taken place yet directly after insertion, right? And sort of the background of this is that whenever you actually look at these de novo insertions and mutator, you find that they have a really, really strong insertion preference, which maybe is a niche in the ecological framework, right? These mu elements tend to insert very, very strongly into the very beginning of genes, the transcription start site in that 5' UTR. And so these red distributions are showing insertions of mutator. The green is showing insertions of the ACDS system, which is another active transposable element system. And you can see just how specific this mutator insertion preference is compared even to this ACDS system, which still shows some preference. This is for the transcription termination site, but it's far less specific than the mutator insertion preference. So the idea with this project essentially is to try to figure out whether these insertion preferences actually represent niches in the ecological understanding, or if they're just insertion preferences that are generated by the availability of loci in the genome, right? There are a couple of possibilities that could explain this, right? It could just be that these are the sites that are available to them whenever they transpose, or it could be that they have specialized for these and it's important for their continued proliferation to target these sites specifically. Another thing that I didn't mention is that they're also highly specific for genes that are more expressed in germinal lineages, right? So this would sort of indicate that there is some advantage to where they're inserting into, right? So the basic premise of what I'm doing is I'm just taking these mu active lines with this active mu-dr elements and a bunch of non-autonomous elements. And I'm crossing them over and over again back to regular B73 without the autonomous elements to see which sort of lineages of mutator elements are actually more successful in terms of creating new copies of themselves. So it's pretty simple, right? I take these active lines and cross them to B73, and then the next generation I take those and I back cross them to B73, and look for which populations are the most successful. But there are a bunch of other things that we're doing at the same time, of course. In this ecological framework, for example, if we're thinking about ecology in terms of organisms instead of transposable elements, obviously something that influences the success of different elements is their environment, right? If there are a bunch of predators of a specific species, whenever it colonizes a new environment, it's going to be far less successful than a lineage that goes into an environment without predators, for example, right? So while we're doing this back crossing over and over again to B73, we're also doing crosses to all of the NAM lines, at least the ones that we could successfully cross to this past summer, as well as a bunch of mutant backgrounds, including, MOP1 and obviously MuKiller at each generation to see how these different sorts of genomic ecosystems affect the success of these elements over time. And, of course, I gave this presentation a couple of months ago when I was in Toronto. So I used the islands off the coast of Toronto as an illustration of this. It's less cool if you're not actually on the island, but you'll just have to trust me that it was cool at the time. Yeah, this is the basic idea. We just sort of look at these lineages over time in different environments and see how successful they are. And the general hypothesis is that those elements that more successfully occupy the niche of mutator should be more successful than those that don't, with success measured in terms of copy number over time, right? So there are a ton of different questions that I'm just going to kind of throw out there that we are intending to address. A big one for me is the relationship between autonomous and non-autonomous elements. Of course, most people assume that non-autonomous elements are essentially parasites of the autonomous elements, right? They don't have the ability to transpose themselves. And of course, they act as sort of a competitive inhibitor for transposase enzymes produced by the autonomous elements. So it's sort of assumed that they're parasitic. And in fact, there's some evidence that generation of non-autonomous elements can even lead to the complete extinction of transposable element lineages over time. But that's not what we see in mutator, really, right? These are sort of somewhat stable lineages or systems of autonomous and non-autonomous elements. So in this system, we can actually look at sort of the changes in copy number of autonomous versus non-autonomous, both due to transposition and genetic segregation. And so we'll end up with some lines that have a ton of a specific non-autonomous element, and some that have fewer of that non-autonomous element, and look to see whether those that have a bunch of the non-autonomous elements are actually less successful than those that have fewer of them, right? There are a bunch of questions that haven't really been addressed before that are just sort of descriptive, like, are there differences in insertion targeting between classes of elements? There shouldn't really be, because they're all mobilized by the same autonomous element, but we'll see. And there's also a bunch of endogenous mu elements in B73 and MO17 and all of the NAM lines as well that we can look at, that we can look for and see if those are also becoming mobilized. And I've already done actually a lot of this with the samples, at least that I've already sequenced from this summer. And it really appears that there's endogenous elements, even though some of them are extremely similar to mu-dr and to some of the non-autonomous elements. It appears that they don't become mobilized, which is really kind of interesting to me because, there's not really a reason that they shouldn't be. mu-dr is perfectly capable of removing epigenetic silencing of its, these identified mu1 through mu18 elements. So it's kind of odd in that way. Another interesting thing is that in these mu-dr populations, or mu populations, they often experience just sort of spontaneous silencing, without introducing mu killer or anything like that. Sometimes they just become silenced. And so these are leaves of a particular plant. These are all the same plant with the less mutation, less reporter gene. And this reporter gene is just a mu-dr that's inserted into a specific gene. And whenever it's active, it creates these lesions on the leaf, right? So you can see that in this particular leaf, mu was very active. There are a ton of lesions all over the leaf. And then if you go up the plant, leaf by leaf, so you can imagine this is looking at the bottom leaf, a leaf at the bottom, the next leaf up, then the next leaf up, and then the next leaf up. And you can see in sectors that there's this spontaneous silencing of the mu system as you go up the plant. So we have all of these different populations of mu, and we can look at which ones become silenced and try to figure out if there's some aspect of mu insertion within these mu populations that sort of tends to trigger this silencing. So I've organized these into sort of mu specific questions and then to questions about the host. Another interesting aspect of this mu system is that sometimes whenever you have a mu active family, plants within that family tend to become what's sort of in the mu system lore known as mu sick. So these mu sick plants, it's kind of hard to explain exactly what the difference in their phenotype is because it's a bunch of different stuff. It's sort of a systemic issue where they tend to be really short and really thin and frail. They're late in terms of producing pollen. This is a kind of, it's hard to, I'm sure you guys know it's difficult to take pictures of corn in the fields because there's corn behind the corn. But you can kind of see that this one's really short and frail compared to this one, which is taller and not sick. And this is the difference between the leaves of those two plants at the same height. But nobody really knows exactly what the cause of this mu sickness is. It seems, just sort of anecdotally like it's related to copy number. Whenever you reach a really high copy number, maybe there's just too many double-stranded breaks happening across the genome that's causing the systemic effect. So we're going to look at that as well, and we just hopefully in December we'll learn about a seed grant that's sort of more specific to that part of the project. There may be populations that are more conducive to producing the sick phenotype than the wild type. So we also want to know how mu silencing affects gene expression. This has been looked at a little bit in terms of just looking at elements or genes that have experienced mu insertion into their transcription start site. But nobody's looked at the expression of the gene before and after silencing, both through the spontaneous silencing and mu killer silencing. So we want to know if silencing the transposable elements has any effect on the genes that the transposable element has inserted into. Those non-autonomous elements, mu1 through mu18, also include small captured regions of genes, maize genes. And there's been a little bit of work done by, for example, Aline Mule in Brendan Gott's lab, or formerly in Brendan Gott's lab, where she looked at crosstalk of small RNAs between captured gene fragments and their donor gene, and she found that there was, generate a large amount of generation of small RNAs between those captured elements and the genes. But here we can actually look at that as a function of copy number of the mu elements, right, and see if there's any effect on expression of those donor genes based on the abundance of their capturing mu elements. Finally, we also want to look at, and this is kind of farther down the line, I still haven't really worked out how this experiment is going to look, but what I'm really curious about is how the success of mu populations sort of depends on different levels of selection, right? You might expect that populations that expand very quickly and produce more copies, maybe they're shorter in the long term, or they're more successful in the short term by producing a ton of copies. But if they sort of immediately get recognized by the host due to their extremely large copy number and silencing, then maybe it's actually more advantageous to move at a lower frequency and just kind of, stay hidden, essentially. But we'll see what that looks like when we get there. So yeah, this is all sort of, as I said, I've really just started. We did a field, field of this over the summer, and I'm right now in the process of planting in the greenhouse to get another generation this winter. And I've 2 more years, so 2 more field seasons and 2 more greenhouse seasons. So there's a lot of, well, there's some time to do these things. And so I guess I'd love any kind of input on things that you think that I should be doing with this, specific mutants that you think would be really interesting to do with this. Before I get too far into it and it's too late to look at it, right? But yeah, that's what I've got for today. I obviously want to thank my advisors, Brandon from UC Irvine, who is my PhD advisor, and Damon, who's my current advisor. These are the other folks who were on this paper that I started with. Alex, Aline, and Edwin, who all did work on this. I want to give a huge shout out to my current undergrad, Alex Osborne, whose LinkedIn I've included here. She's fantastic. So, in a couple of years, if you're looking for a graduate student, remember this name. She's great. She's done more work than me on this project, honestly. So absolutely look out for her. And the besieged staff of the National Science Foundation, especially Shinhan, who helped get my current postdoctoral fellowship across the finishing line. And yeah, that's what I've got.
Nice talk. Thank you, Galen. So we have quite many questions in chat, and I think we can start from John Hodge. John?
Yep. No, it's a really, really interesting topic. I guess I was just kind of thinking, I tend to think about like evolutionary patterns among like synthologs and homeologs and how you'll sort of get these punctuated bursts of evolution in like the level of the protein. And when you were sort of talking through these like highly conserved or these like highly structured elements that can form these hairpins, it kind of has me wondering, uh, have do you have any sort of intuition like how they're sort of arising and if they might actually sort of decay out? Like, is it just sort of like some random evolutionary walk? Does it look like it's some sort of like punctuated process where the structure isn't there in like one generation and then suddenly it is? I guess for me, it's really interesting that they're there, and it's just like, well, why are they there?
Yeah,
Uh,
yeah. Well, yeah, it's difficult to say. We looked at centenic regions of gene or centologs between maize and sorghum, and we didn't specifically look to see if the centilog in sorghum had structure, or the same level of structure, which I wish that we had. But there's also a distinction here that I haven't made between like exons and introns. And certainly structure can arise extremely quickly in introns if there's, for example, a highly structured transposable element that inserts into that intron. We did a bunch of analysis on like to see if there were similarities in sort of like DNA motifs, for example, sequence motifs in structured regions between genes, and none of it was super convincing. But yeah, I actually didn't look, I guess, that much into how they arise, but they're frequent in both exons and introns, and yeah, um, I'm not sure if that addresses your question, but yeah,
All right. It sounds like it's just there's a lot of work to be done, but no,
yeah,
thank you.
for sure, yeah.
Yeah, good. Jeff, would you like to
Yeah,
ask?
sure. I had two questions here. It looked in your when you were looking at the skew across superfamilies, it looked like there was at least one DNA superfamily where 24mers didn't show skew or showed
Yeah,
less skew.
yeah.
Any idea why?
I have absolutely no idea why.
Cool.
Um,
Okay. That makes me feel better.
yeah, yeah. I feel like, this was a while ago, so I don't exactly remember. But I'm sure that we said something about it because it is like, yeah, there's like one fam or super family where that's the case, and it's a bit odd. But yeah, I
And
think
then
that those also just had fewer structured regions as well. But
yeah,
that's what,
okay. And then you looked at variability and expression across the NAM. Is there a variability for how miRNA like miR whatever miRNA like the DNA sequences across NAM?
that is an excellent question, and if I'm remembering correctly, I think that the deal with this was that this NAM analysis essentially came between submissions of the paper, uh, and we were really trying to get it out. So I don't think that we actually looked at that, but that's something that I'm doing now. In fact, I know that we didn't look at that, but that's something that I'm doing now with Brandon is actually looking at structure across all of the NAMs and other species to see how variable it actually is, evolutionarily.
Cool.
So I don't know.
Um, and then I know Ed has a question, but you proposed that there's this causality of mRNA-like structure to higher and more variable expression. Could the causality be in the other direction, that genes that are more highly expressed and more
Yes.
variable? Yeah, okay.
Yeah. No, I absolutely think that's probably, uh, yeah, yeah. It definitely could be the case. And if you read the paper, this is obviously the presentation form where I don't want to get too into the weeds, but it's a little bit strange, this variation in expression, because we also looked, for example, at like variation of expression across tissues within lines and didn't find that there was higher variation in the structured elements. So yeah, I'm very cautious to say that it's actually causative, um, for sure.
Thanks.
Yeah.
Next, Ed. Ed here.
Yes. Yeah. So, Ed, is your question the one that you've posted in chat? I wonder if these structures exist in the DNA, not just RNA. Do gene hairpins have the same variance in length as TEs do? Yeah. So this is also something that I'm working on with Brandon right now is structures in the DNA. Um, as I showed in that sort of last figure on this project, we've basically just divided the genome up now. Whenever we did this project, we were just taking the sequences of these annotated elements and running them through these pipelines and looking at structure. And what I'm doing now is dividing the entire genome up into windows of varying sizes and looking at structure within those windows. Because yes, there certainly could be structures in the DNA. For example, we find a high degree of structure in knobs, which are not, to my knowledge, at least I don't know that much about knobs, but to my knowledge, not expressed. Um, and yeah, of course, there could be interactions, for example, with recombination and the structure of the DNA preventing recombination or increasing recombination. But yeah, I'm not sure. Yeah, there's essentially it's really difficult to distinguish whether the importance of the structure is at the DNA level or the RNA level, right? Unless you know that something is not transcribed.
So, Galen, I'm curious about the impact of the structure, helping structure on the protein
Translate.
translation. Yeah. Could you take a look at the mRNA and the protein difference and then the expression difference and see how that impact upon the structure?
Yeah, I would very much like to do that. I've not done it. But that was sort of the implicit, I guess, argument for why a lot of genes might have a high degree of secondary structure within exons, like particularly the UTRs, for example. It's, there's literature from a long time ago suggesting that the structure of the transcript is really important for loading into um, ribosomes. So I'm sure that there's some relationship between the hairpins and translation, but I have not looked at it specifically with this data.
Thank you. Ellie, would you like to ask your questions? Maybe you have two questions here.
Yeah, thank you. This was a really cool talk. I am going to start a postdoc with Brandon soon, so this was
Oh,
a really
nice.
good recap for me of some stuff
Congratulations.
that I'll need to read more about in like a
Yeah.
month. And I just have a couple questions. These are probably in the paper, so you can just tell me to read the paper if these are answered there. But one thing was a lot of the TEs and genes had higher signals at boundaries. And I think for some of them, it's like only at the 5' or 3' end. So maybe for those, the signal is really clear. But I was wondering, like, presumably for mRNA, the edges are more flexible and more dynamic and can form the hairpins more easily.
Mhm.
Then how do you tell the difference between like a computational boundary effect at the edge of the mRNA and like a real effect of the hairpins forming more easily there? Like, is that part of the structure fiction?
Well, yeah, I'm not sure if I totally understand your question, but uh, my intuition is that the computational effect would be to reduce the amount of structure that you would see, um, because you have these windows where you're cutting things off and maybe uh, not allowing inference of base pairing between things that actually would have base pairing between them. I can tell you, and actually maybe I can show something here because this is obviously something that should be, if that is an issue, it should be ameliorated by what I'm doing now because I'm not specifically looking at genes that have been cut out of the genome. I'm looking at windows that happen to contain genes and contain the
Oh,
boundaries.
yeah, right.
And
Yeah.
so it is really interesting, actually, and I'm sure that we'll meet and talk about this at some point in the next couple of months.
Nice. Yeah.
But if you look at genes, if you look at those windows across chromosomes, and you look at genes and you look at transposable elements, both of them actually tend to sort of get uh, like a kind of odd increase in secondary structure right before the genes, including in windows that don't include that part of the gene, which is odd because, you don't expect those to really be transcribed, right? Transcription is
Mhm.
assumed to start at the transcription start site. So it's odd that those have any kind of signal like that at all, but it's what we find. But anyway, yes, we still find, even in those sort of windowing approach that doesn't just cut out the gene, we still find this signal of high structure in those UTRs.
Yeah, so interesting. I guess that's a very distinct like niche habitat right in front of the gene.
Yeah,
Like
yeah, yeah, yeah.
maybe some like um messiness or like gradient of what gets transcribed, and like the transcription start site might not be as clear cut.
Right.
Yeah.
Absolutely.
Okay, so thank you for that. And then the second question was really just quickly developmental. I was curious if you checked whether, like, I know some mRNAs are important for development of like leaf primordia or like setting up different
Yeah.
developmental zones. I was wondering if any of the genes that you found that have like the mRNAs are genes like that, and you could check what categories those genes fall in to see if the variation that you don't see within tissues, but like between lines and across tissues might
Yeah,
affect development.
I think that's an excellent idea. And if I could do it over again, I'd barely seen a corn plant in my life whenever I did this project. So it was all very abstract to me. But yeah, I think that's absolutely maybe a next step is to look at it, because we did look across tissues, but only really in terms of this variability. And we found that actually these structured genes tend to be less variable among tissues than the non-structured ones.
Huh.
So, but I don't necessarily know if that would mean that they're less differentiated among tissues. I'm not sure.
Yeah.
Yeah, there's a lot more to do with this. A lot more.
Well, thank you.
Yep.
I would like to jump to the John Farrell for the last question. Okay.
Hi, thanks, Gayla. That's super talk, really interesting. And I guess this a little bit follows up from a little bit of Ellie's question and sort of thinking about developmental expression patterns, and I was curious on your sort of your variability across the NAMs, how you took into account the developmental expression patterns of genes, and I have a little bit of a bias here, and I'm in that I'm most interested in pollen. And so are there any particular, sort of pollen expressed genes are more or less stable, sort of in the variability you saw? Um,
Yeah, that's a level of granularity that we didn't really
mhm.
get to, because we essentially we looked at variability of expression among tissues and variability of expression among the lines, but we didn't look at variability of
Yeah,
among tissues among the lines, I
among
guess.
the lights. Yeah, yeah, yeah. So
Um, yeah,
there are some suggestions that pollen has sort of differences in how it deals with
sure.
gene expression and epigenetically. So that might be worth
Yeah,
looking at.
yeah,
Um,
yeah, yeah.
and then the other sort of the final sort of point is, you mentioned that that these insertions are not necessarily influenced by selection rapidly, but I would just note that gametophytes get selected on in as haploids potentially in
Right.
the first cross,
Mm-hmm.
and uh, we have some evidence that that genes that are expressed in pollen, particularly ones important for pollen fitness, are you don't you recover heritable insertions in those at a much lower frequency.
Right.
And I
Right.
was wondering if I mean, it sounds like you're just starting the project, but have you seen that? And if you haven't, I'm very interested if you do see that.
Yeah, no, no, I've not gotten that far. But yeah, that's a really great thing to be thinking about, and I appreciate it. But no, I haven't gotten that far. I'm really still in the process of just sort of like figuring out which primers to use to make sure that I'm recovering all of the
Right,
active elements.
right, right,
Yeah,
right. No, well, it sounds like a cool project, so good luck.
thank you, thank you,
Nice discussion. Thank you all the questions, and thanks Galen for the nice talk,
thank you very much for having me.
and thank you all for coming to the seminar. And we'll come here next week for Alice talk.
Great,
Bye.
thanks everybody.